Welcome, welcome, cell biologists. We have just finished talking about chapter 18 when we discussed the first part of gene expression, which is transcription or making mRNA from your DNA template. Now we're going to get into the second part of gene expression, which is translation or using the genetic code to take that mRNA and make proteins. We're going to start by just kind of overviewing this concept. Then in the next video, we'll talk about the different molecules that are involved, and then we'll get into talking about what's actually going on in protein synthesis. So some of these genes that we made RNA, and sometimes that's the end. That's as far as you have to go. But for most of your genes, you are going to have to take that RNA and use it to create a protein. That ultimate functional product is going to be a protein. So the mRNA, which is carrying this code from the DNA, is going to have the instructions for how it needs to be used for translation to make a protein. In translation, we are taking individual amino acids together, stringing them along, and making a functional polypeptide that'll fold into the specific 3D structure to have a working protein. So the way that these DNA bases from the RNA can be used to make a protein, which is a completely different language. Those rules are called the genetic code. And I'll show you how to use the genetic code, but I definitely do not expect you to memorize the genetic code. You need to be able to figure out, with a copy of the genetic code, you can figure out what the protein will be. You can figure out where it'll start and stop and all of that, but you don't have to memorize what makes each protein. There is a theory that is labeled the one gene, one polypeptide theory. And in general, that means that in one gene, you are going to end up with one polypeptide. So you have a gene here in your DNA. Your DNA, your long chromosome, has lots of genes on it, but one gene is going to be used to make an RNA product, and that RNA is going to be used to make one polypeptide, which would fold into a protein. There are some situations where it's a little bit different than one gene, one polypeptide, but for the most part, that's how it works. When you have one gene, it's going to give you one polypeptide. If you change your gene, you're going to change the protein you make. So a mutation in your DNA, a mutation in your RNA is going to give you a mutation in your protein. Our genes are written in a language of 3-letter words. So here is our, pretend like this is your DNA, 3-letter words on here. And if you know how to space it correctly, you know that each word is 3 letters long, you would say, our big red dog bit the old man. But what if you have a mutation that inserts something or deletes something? That's going to be an indel mutation, an insertion or a deletion. That means that with one insertion here, just stick an extra X in here, you've suddenly now made this language be our you've made complete foolishness, right? Like, your cell is not going to make that into a functional protein. What if you delete something here? Now, by deleting it, your words are now saying our, ed, agb. Again, complete foolishness going on, whatever it's doing here. So you're not going to get your functional protein. So single mutants, indel mutations, insertions or deletions create mutant proteins. And these are called frame shift mutations. So even though the rest of these letters are here in the right order, you've shifted the frame. So, and you've changed from being able to have the right big and red words being written here to suddenly you're breaking up your words in the wrong order. You've shifted the frame from where you're reading them. Our DNA, of course, isn't going to have all of these letters making words like this. Our DNA is going to have A's and C's and G's and T's that are going to create 3-letter words to make proteins. So if you take, you say there's A, C, G, or T in the first of that 3-letter word, you have 4 letters that can be the first letter. You have 4 letters that can be the second letter. You have 4 letters that can be the third letter. 4 times 4 times 4. That gives you 64 triplets that could be made out of those 4 nucleotides. So 64 different combinations we have 20 proteins, or 20 amino acids to make our proteins with. That means that of those 64 codons, or 64 nucleotide triplets, some of them are going to be used more than once for each amino acid. So that is what, when you hear the genetic code is degenerate, that's what it means. Some codons, some of those combinations are going to have the same meaning. So it would be like saying big and huge have the same meaning. In your codon chart, different codons will have the same protein. And the protein, or the codon sequence, is non-overlapping. So here, if our sequence is AUG, GGC, UCC, we make AUG the first protein, then GGC is used for the second protein, and UCG is used for the third protein. There's none of this overlapping mess going on. So those 3 letters in your RNA, that triplet is called a codon. And your cell has transcriptional machinery that will read those RNA triplets. And the codons are always going to be written in a 5' to 3' manner. When we were starting to figure out how to figure out what is in our genetic code, a scientist took a system like out of the cell, just took all the components with lots of U's. So they didn't have the C's and G's and A's weren't in there. Lots of U's. And when they just made RNAs with lots of U's, they had a polypeptide with lots of phenylalanines. So they were able to figure out that UUU encoded for the amino acid phenylalanine. And then they kept figuring out, they made the same system with all adenines in it and figured out that AAA codes for lysine. And then with all just cysteines in it, they figured out CCC codes for proline. And then they started to make copolymers where they're mixing 2 different kinds of nucleotides. So they do something where your RNA is something like UA, 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 UA. So that means you're going to have 2 codons, UAU and AUA. And figured out those 2 give you either tyrosine or isoleucine. So we know one of these is tyrosine or isoleucine. Kept doing these experiments again and again, mixing up what are our different codons that we have. Eventually figured out each of the codons gives you each specific amino acid. So here are each of the 64 possible codons from UUU, UUC, UUA, all of these. And remember saying that it is degenerate. So here you can see UUU, UUC, both of those codons are read as phenylalanine. These mean exactly the same thing. They're phenylalanine. All of these codes will give you leucine. So those are all synonymous to each other. They all end up with leucine. There's, of these 64, 61 of them will give you a specific amino acid. One of them is really important, AUG, which is the start codon. It'll cause methionine to be put in there, but that is the start codon. That means in your big old piece of RNA, your translation machinery is going to look for that AUG, and that is where translation starts. And translation is going to keep running along until you get to one of these stop codons, UAA, UAG, and UGA. At those 3 codons, we don't put in another amino acid. At those 3 codons, it triggers the entire translational machinery to just fall apart. That is the end. It's stopping translation. Most everything uses this exact amino acid. Whether you're a bacteria, an algae, a fungus, a plant, a person, this is really what's used. There's a couple of minor changes. So like in mitochondria and some bacteria, AGA is going to be used as a stop codon instead of an arginine. There's a few non-standard amino acids. So, methionine could be a different modification of methionine. Some of these amino acids are going to be modified a little bit, non-standard. But really, the genetic code is almost universal. Everything that's alive, everything that has DNA, makes RNA, their genetic code is going to look mostly like this. And then while we're talking about the RNA being degenerate, there is also codon usage bias. So that's when you're saying, okay, all of these codons will give me valine, that doesn't mean they're each used 25% of the time. One of them could be preferred over other synonymous codons. One of them could show up more, some of them could show up less. All right, I am assuming you already know how to use the genetic code. This is a 3000 level class, so I'm assuming you can already, if I give you a piece of DNA, you already know how to figure out what the RNA is going to be, and you can already figure out what the amino acid is going to be from that. If you need to, you can go back into some of my other classes. I'll link in our D2L page that shows you how to access some of those videos about how to use the genetic code so that you can be a little more comfortable if you're not. All right, I'll see you next. We're going to talk about what are all these different things we're using, how, what amino acids are, what's an mRNA, what's a tRNA, how is the ribosome made. So I'll see you in a little bit for that. Bye.